Fusion, the International Science Radio Show.
I'm Professor Liz Harry from Emeritus Professor at UTS, University of Technology, Sydney. I am also a Senior Associate at Outside Opinion, which is an organisation that gives expert advice and opinion on anything to do with academic research, really.
Most people have an idea that bacteria are one of the microorganisms that can cause disease, but there's a lot more to bacteria than that. Where do we start the secret world of bacteria?
Yeah, so most bacteria don't cause disease. In fact, we wouldn't be here if it weren't for bacteria. Um, they have been in this world for approximately 3.7 billion years. And we've probably, as modern humans, have come onto the earth in about 200,000 years ago. So these bacteria didn't even have hosts per se. They just survived. They are so diverse, they can live pretty much anywhere. So they're just minding their own business and surviving. And then we came along, and some of them like to live on us and in us. And as a result of that, some of them cause disease. But most bacteria on us and in us are actually, we still also can't do without them because they provide some protection for us. So most of them don't cause disease. I would say, the vast majority.
So these bacteria are absolutely everywhere in every niche, I understand.
Yes, yes. They inhabit extreme temperatures, hot and cold, extreme salt, extreme acid, alkali. You'll find them in the bubbling sulfur pools of, Rotorua in the bubbling mud there. You'll find them in Yellowstone Park, which is very famous for its bubbling sulfuric um, pools. So very hot there. You'll find them between ice layers in the Antarctic. You find them in the gut, and you find them in, a termite gut and a koala gut is very different to a human gut. So they can adjust. And I think because they've been around for so long and because they're so diverse in every way, particularly their metabolism, they can pretty much, different ones can metabolize, i.e. eat and get energy from different types of molecules, from acid molecules, from organic molecules to sewer and also our own nutrients. So they can decompose things, they can do all sorts of things. They can eat oil. But because they're so diverse, it's not all of them can do everything, but they have their niches. Yeah.
Hmm. So eating oil and maybe eating plastic and other things we don't want to keep around.
Yeah, actually, I think, when was it? Not so long ago, I think it was 2016 in Japan that it was discovered that there's a bacterium, and we're continuing to discover bacteria. There was a bacterium that could actually eat plastic that's found, I think it's the PET bottles plastic. And so there's a search for other bacteria that might eat other plastics, which is, amazing. Yeah.
Particularly the ones, I mean, PET we can a little bit recycle, but there's so many we can't.
Yes, yes. So that would be great. I mean, think of all those plastics in the or the microplastics in the ocean, that are just being swept into a big heap.
Yes.
Imagine what we could do with that. I don't know if they're land or water ones, but there will be, wherever we look, we find more bacteria, more viruses, more microbes.
More and more and more. And how
Yeah.
long have we been looking for bacteria or at bacteria?
How long?
Well, I believe, what was it, the invention of the microscope or?
Yeah, so it was in the 1600s. The first person to see microbes was Robert Koch. Um, he um, he developed a very primitive microscope, and he saw molds. That's what he was looking at. But only some years later, Anthony von Leeuwenhoek, I don't know how you say that, it's a Dutch name,
Sounds right.
also was inventing very primitive microscopes and saw these little moving creatures that looked like one little unit of what we now know as a cell, and he called them animalcules or something.
That's it.
Yes, I think, yeah.
That's what I've seen.
Um, yeah, and at that point we didn't know what they were and we didn't know much about them at all. And then later on, we Koch, I think, showed that they cause disease, these things. Yeah, some of them. Yeah,
Some of them, as you say, that's
yeah,
right. And
yeah.
so things have gone on from there where we've been able to look closer and closer at what they do. And we've been learning some of their tricks to do
Yes.
things like genetic engineering.
Yeah, yeah. So genetic engineering with them is, that's the sort of in my PhD, that's what I did. I was interested in biochemistry, cell division, and how these things happen. I'm very visual, so looking at cell division was very interesting. And they have the most amazing ways of doing that. And in order to study bacteria, one of the great things is you can manipulate them genetically very readily. We tend to use model organisms to do that. Many of the model organisms don't cause disease. Some of them do, but we can put DNA in there. We can make them express certain genes or make them not express certain genes under certain conditions, and see what happens. So it's very easy to do this. And I guess, the Nobel Prize, I think it was in 2020, the Nobel Prize was in chemistry, I think it was in chemistry for CRISPR, which is
Mm,
a type of thing bacteria have that gives them a sort of adaptive immunity to viruses. And this is just such a recent discovery. I think the first paper came out in 2006. So, bacteria are not done and dusted. That's the important thing. But we have now adapted that CRISPR system to be able to be used to do gene editing. And it's hoped that this will be used in vaccinations, but it's also looked at for cancer treatment and a whole lot of other things because it's a very quick, easy, efficient way of changing the DNA sequence. Yeah.
mm. And
Yeah.
changing it in the way you want it to be changed only.
Yes. Yeah. So how we do recombinant DNA work is often using bacterial mechanisms,
Mm.
right? So we can make bacteria do things with that, but we can also make other genes do that. Yeah,
It's
in other organisms. Yeah.
it's so powerful, isn't it? Because you can not only can you do basic science to understand more about the genes and what they do, but you can direct them to the places you want to make things.
Yes. 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 It's quite extraordinary. We can really manipulate a lot, if you like, and that makes it so easy. I mean, bacteria grow so quickly. Ones I studied, I always make a joke that I work with bacteria because I'm impatient. But the bacteria I studied, they can double in 37 degrees. They can double in 10 minutes.
Brilliant.
So that's pretty clever to be able to do all that in 10 minutes. Yeah, so yeah, and I think obviously we'll find more and more. I always, when I used to lecture at university, I would say, you know, I'll say this today, tomorrow you might find a paper in Nature that says exactly the opposite. And while it might confuse students, it's important to understand that it's the thinking and the discovery around anything in research really that enables you to realize the dynamics of it. And it's really the thinking and the innovation that matters. Yeah.
Yeah, yeah, there's
Yeah.
so much going on there. And
Yeah.
is that related to the technology that was used to make the COVID-19 vaccines?
Um, it's a good question. I think I think at the moment, no, CRISPR wasn't used for the vaccines, but they're looking at that now. But I don't think they necessarily use bacteria to do that. They could have used it for some of the cloning, but they're mainly using cells and animals. Yeah. I mean, I never expected the COVID vaccine to come at all necessarily. I mean, most vaccines that we try to make, we have never made them.
Hmm.
Smallpox is the only one that we have actually eradicated a disease pretty much, although. might speak too soon. Um, yeah, so the fact that we came up with not just one, but quite a few is extraordinary. Yeah. And if you ever get a chance to read Vaxxers, which is the AstraZeneca story, it's fantastic.
Oh, wow.
Um, yeah, it gives you a scientist's account. I think there are two women in Oxford who give they write alternate chapters, and they just say what they were doing at the time and bring in the work that they had been doing and how it relates to the discovery. And they even put what the formula is for the vaccine,
Goodness.
exact things that they put in it, so people understand. Yeah, I think it's great.
You know who
Yeah.
wrote?
Oh, let me see.
Just
Do you
out of
mind if I have a look?
I have no, go right ahead.
It's
I've just
Catherine,
heard of it. That's all. It sounds amazing. Sounds like a must read.
Catherine Green and Sarah Gilbert. I think it's fantastic.
I
And
will.
I know that AstraZeneca got a bit of a negative thing with the problems with the
Yes,
heart side
the
effects.
vaccine. Yes.
Um, but there was also, I think, my feeling is there was a bit of politics around the US versus the UK. Pfizer was in the US, so there would have been a bit of competition around. But yes, I certainly had the AstraZeneca vaccine because it was the first one available, and I was very happy to have one at that point. Yeah.
Well, I think a lot of people are very happy to have
Yeah.
this in.
Yeah. I don't think people realize how much it saved. It could have been an absolute, it killed a lot of people,
That's
but
right.
it could have killed a lot more. Yeah.
Exactly right.
Yeah.
Back to the bacteria. So how long is so you're saying they can reproduce as quickly as in 10 minutes at 37 degrees. So is that a generation for bacteria?
Yes. So some bacteria do that. Of course, 37 is not the best temperature for all bacteria. It is for the ones that live on us, and it is for some others. Like I was studying a soil bacterium, Bacillus, and uh, it's not a germ, but it's a wonderful organism to work with. So it divides very quickly, yes. And what we know now, and I've been studying the cell cycle, so multiplication is my thing. And yeah, so in 10 minutes, what we decide is a generation time, I guess, what we define as one is one where the mass doubles. What's the time in which the weight of the bacteria doubles? And what we usually measure that using absorbance at 590 nanometers. That's what we do. So in the visible light. And it's the turbidity of the actual medium that you look at. So you can study that and you can see that they grow what we call an exponential growth. And yeah, that's the 10 minutes. So to do that, yes, they have to grow quickly. So they have to make more envelope to make them longer. And this rod-shaped bacteria, they usually get twice as long. What happens in the middle, though, inside the cell is very interesting because you've got generally bacteria have one copy of DNA. They have one chromosome. They then have to replicate that chromosome, separate those chromosomes, and then a division occurs between the two chromosomes. Now, to do that in 10 minutes is not possible. So what they do, because the whole chromosome takes about 30 to 40 minutes to replicate in these guys. So what they do is they start a round of replication, and then before that round is finished, they start another one. So you have these overlapping cycles of DNA replication, and everything else is going really fast. But that's the limiting step, really. And so, in terms of control, it's a researcher's nightmare because it's very hard to tell what's controlling what because you've got so many things happening at once and you don't know which cycle you're studying, right? Because you've got multiple cycles going on. Um, and you've also got chromosome segregation happening. So when that's actually starting to happen, we know now as soon as replication is started and the beginning chrome of the chromosome starts to move apart, you've got the chromosome segregation proteins doing their thing. This is very different to even the simplest of eukaryotes, the yeast cells and our cells, where and many people have seen this. You have a round cell, you've got a lovely mitotic spindle with chromosomes lined up. You get replication, then they line up, and then they separate into their copies of chromosomes, right? And then you get a division. So you have to, that's how you have to have these cells, these more complicated cells. Bacteria, it's all happening. And there's also no compartments in bacteria, so it's hard to study. So how they do it exactly in 10 minutes is hard to know. What we have studied, though, is I studied this organism because it forms spores. And that's just a dormant round cell that is inactive. It's inert, like a fungal spore, like a mushroom spore. You know, it's very, very similar. If you get a lot together, it's like a whole lot of brown dust. So when we put that into a certain medium that enables it to germinate, just like a seed, that first cycle you can look at in terms of regulation because it's the first one, doesn't have any information from the previous cycle, so you can do things to it and see what happens. So we did that because you have this lovely, and it's quite synchronous. So we did that and worked out that the division, the first step in division is coordinated with the first step in DNA replication. Yep. And we started to identify molecules involved in that process. And there are a lot of people working on cell division still now to work out how it's coordinated. Yeah.
And the other thing there is that you often get an idea in basic biology classes that that bacteria are just asexually reproducing and making copies of each other, but there's actually some genetic exchange going on, is my understanding,
Mhm.
right?
Yeah. So, yeah. And again, this is quite late. And I think, look, I think it might be a Japanese guy that firstly discovered this. And I think he was sacked in the end. It's a great story that some very famous biologist told me. And anyway, people didn't believe it. And this is the thing in science initially, and it's sort of a safeguard, so we don't just believe everything, right? That we hear. But we can take it too far. We don't necessarily want to believe the world is different to how we understand it. Yeah. I mean, that even with scientists, even though we love discovering new things, we don't like to be told we're wrong or we haven't got all the information. But of course, that happens all the time. So, yeah, this guy discovered what he thought were mobile elements and that genetic exchange was going on. I think he got a job in America eventually. Yeah, so this is a whole new world where bacteria can take up naked DNA. Some of them can take it up just if it's in the environment, which is how they can be so diverse. They just change DNA all the time. So there's various ways in which they exchange DNA. But one of the most interesting is one called conjugation, where there's famous pictures where you've got a tube forming from one organism to the other, and it can be the same species or different species, which is quite amazing. And the DNA goes through. So the mobile DNA goes through. It can replicate itself. It can take on so many different forms. It's ridiculous. And it can also be transferred. So, people always joke about bacteria having sex through this little pili, which is what it's called. That's a bit of a push, I think, but anyway. So, what happens then is that bacteria takes on whatever genes that are in that mobile element. And they could be for antibiotic resistance, they could be for adapting to anything, like it could enable them to metabolize oil, for example,
Mm,
right? So it changes them, which is good, but it's very bad for antibiotic resistance. I'll say that right now.
mm,
Yeah, so, and what that means is that you can get that exchange of DNA without division, right? So it can just happen. So it happens very, very quickly. And what that means for the world, I guess, and our understanding of bacteria and evolution is that gone are the days where we just are looking at the chromosome because the mobile elements are working with the chromosome and in addition to the chromosome to actually change how bacteria feed, how they might adapt to something, could change their shape, could change their defense to antibiotics, defense to immunity, all that stuff.
and
Yeah,
when you
so quite
say
profound.
it's very profound, and when you
Yeah.
say different species, is that just different species of bacteria, or can they pick up genes from other organisms that are not bacteria?
Not that I know of. They can pick up other. Yeah, not that I know of. Um, sorry, they can pick up viruses. Yeah. So sorry, I wasn't thinking about viruses.
Mm,
Yeah. So the same with our cells, we pick up pieces of viral DNA. So that also has an effect on our gene expression and what we express from our DNA. Yeah. So viruses in general inject their DNA, and there's RNA viruses as well, often by just um, attaching themselves to, some of them look like little spaceships, and they attach themselves to the cell envelope of bacteria, for example, and they'll inject their DNA. Yeah. And sometimes they will just go into a phase where they just insert their DNA into the chromosome and stay there and be replicated along with all the cell divisions. Yeah. Other times they will go in and they will go into what we call a lytic cycle, which means a cell bursts open with 100 million viruses or millions of viruses. And the viruses have got the genes to undergo that cycle, but they also use some of the host genes to replicate, right? So you've got a lytic cycle and what we call a lysogenic cycle, where they just sit there, depending on the conditions, right? They're clever little selfish genes, as Richard Dawkins calls them. Um, and they can also go from that sort of dormant phase where they just hang in the DNA of the host into the other cycle. Yeah. So when we get a viral infection, the same thing happens. Goes in, multiplies up, makes more virus with some of its encoded DNA. That's it. The coat of the virus is usually made of protein and other things. And then there's so many of them, and they cause the lysis, which is the bursting open of the cell. And you've got millions more, and they go straight back into more cells. Yeah. Yeah.
amazing. Because I
Yeah. Yeah.
thought I so the viruses are also taking DNA from their hosts sometimes, and
Yeah.
they're sharing them with bacteria. So the bacteria could end up with genes that were originally perhaps in animals or plants.
Yes. Yes.
Amazing.
Yeah. Um, Most viruses, though, have specific hosts. So, as you know, there's ones that we could transfer from birds to us and so on. So, the
Mm,
flu ones, but they're more related to us than bacteria. So, as far as we know, the bacterial viruses are quite different to the higher organism viruses. Yeah,
mm,
but I think there's probably a lot of data on this, but I don't know much about the exchange of viruses exchanging DNA between different species and between quite different organisms.
mm,
Yeah,
mm. Oh, it's one
yeah,
of those things. So it's
yeah,
just so the implications for evolution, as you say, so one, you can watch evolution happening because bacteria reproduce so quickly compared to
yes,
animals and plants. But then
yes,
you've got all this sideways transfer happening as well.
yes. And it's really, well, you could call it a headache or an exciting time for a taxonomist, to sort of think, well, we can classify initially before we had DNA sequencing, of course.
Mm,
We could classify bacteria on shape, perhaps conditions under which they survive. They could be aerobic, which means they like oxygen, or anaerobic, where oxygen is not good for them. They could be photosynthetic, where they make their own food, like cyanobacteria or whatever. Maybe where they live. Um, do they have appendages? And there was always this gram positive, gram negative stain that people used because bacteria have a cell wall. So
mhm,
they have their envelope is the envelope around their cell is very different to humans and other organisms, similar-ish to plants, but they have this very rigid cell wall that gives them this shape that they have. They can be spiral, round, long rods, short rods, and anything in between. So that cell wall allows them to change their shape. Again, it's fairly new discoveries. And the difference in cell wall structure has been used. So some of them have got very thick cell walls, which is called gram positive because it stains differently under microscopes with certain stains. and then you've got the gram-negative ones. Yeah, so they have a different cell envelope, and so that changes. It changes some of the division machinery and some of the other things that they use to keep the cells safe inside from the outside world. Yeah, yeah.
So I guess in addition to looking at just how bacteria live and how they operate with us, they're also a big part of our environment.
Yeah, they're everywhere. They're everywhere. So, um, I guess inside our body and outside our body, since we've been able to sequence and been able to look at microbiomes, particularly the gut is a very popular one. It's hard to look at the human microbiome, of course, unless you can take samples. But skin, gut, vagina, any oral, all sorts of, different areas. We now know that this microbiome is extremely important, and the gut is another field. However, it's very important because whatever we eat depends on is changes our microbiome, and the microbiome itself is needed to digest food, and it's needed also to keep the bad microbes away, so the ones that cause disease. Okay, so yeah, in our environment, if you just talk about this environment, even on the skin, there's microbes all over our skin, and they keep fungi and various other bad bacteria from getting there, which would make our skin go dry or maybe cause infection or make it be itchy or whatever. So all those sort of immune responses that we don't have to bother with, most of us. I know there are still some skin problems with people, of course, but we wouldn't survive if we didn't have them on our skin, pretty much. And the interesting thing then is they have to be in those places. Infection then happens when you might cut yourself, and then the beautiful bacteria that are protecting our skin go inside. That's why it's an issue. Yeah. So in our body, yes, very important. Nasal passages is another one. They're there to protect us, and we were never without them. We've always been with them. Helicobacter is something that is co-evolving with us because it is in the stomach. That's where it is, right? It's really interesting evolution stories. Then you've got bacteria in the soil. Of course, there's a lot of talk about agriculture and crops and good soil, when we didn't know about this, of course, colonization and all the exhaustion of soil is partly because the microbes are gone. I mean, there's zillions of microbes in the soil, and they're all happily, if you just a healthy soil is where you've got buckets of microbes, buckets of worms, and everyone doing their thing, decomposing, releasing nitrogen, oxygen, and all sorts of nutrients for plants, for the animals to eat. Okay, then you get healthy animals, you get healthy vegetables, and so on. So yeah, it's lovely to see people doing this. It's obviously a challenge because it's a change, but also, you've got to spend a bit of money doing this. But people are doing this with crops now, moving away from just exhausting the soil, because otherwise we end up with parts of land we can't use. Yeah. But
That's
we can revitalize them. Yeah.
so by growing the bacteria in the soil, we're growing the soil.
Yes, yes. And of course, then you have bacteria in the ocean that
Mm.
people feed on, organisms feed on. I mean, everything, back, everybody eats bacteria, pretty much. Yeah.
And how did you first become interested in bacteria?
Well, it was sort of the molecular biology era. It was the 80s when I did university.
Mhm.
And molecular biology was a new thing. There was one molecular biologist in the biochemistry department at Sydney Uni, which is where I did my degree. And there was one molecular biologist in the biology, they were separate buildings. And I liked genetics. I really liked genetics. And so I chose one of those people in biochemistry, and both of them actually worked with bacteria. And that was a nice time to do molecular biology with bacteria because they were quite, they were the organisms that you could do it in. That was where it was discovered. And sequencing was tens of years that sequencing had been discovered. So it was quite new. And this guy happened to be studying bacteria. His name was Jerry Wake. He was a professor, fantastic, fantastic, bright, excellent thinker, and a very, very lovely man to work with. So I worked with him doing honours, and he was studying termination of DNA replication, so how the chromosome separates and what happens at the end. But he'd had a student some years ago that was studying cell division, and he sort of put it on the shelf for a while, and he said, look, I really want to bring it out again. So I was interested in that because you could see division, and I've always loved microscopes. I've always just loved seeing biology. So bacteria was good for me. As I said, I'm impatient. And so they double very quickly, and you can, grow them up overnight, grow them up in the morning, get them to exponential phase, shove them under the microscope and have a look and see what they're doing. So I discovered a couple of genes involved in cell division. We didn't know at all what happened to divide the cell. We could see it with certain resolution microscopy, but we didn't really know how it worked. So I cloned some of those genes, if you like, using very beautiful, simple methods, but not what you'd use now because a bit too slow. So for my honours, I did that. For my PhD, I identified what products they produced, and those proteins are still being studied. And one of them actually is an attractive antibiotic target. And we now know, always thought the bacteria didn't have a cytoskeleton, but it turns out that they have an actin homologue and a tubulin homologue, and both of those are found in humans. One of the proteins that I discovered was actually turned out to be a tubulin, the precursor of human tubulin, which is, years and years later that was found. Yeah. So
what does tubulin do in humans?
So tubulin in humans, its major role is to form a spindle. So the cell is an oval shape, and the spindle is hooked up on either side or the poles of the cell, and I guess it's a bit like a muscle, or it's like a skeleton, and it lines up. The chromosomes attached to that, and they line up in the middle. Okay, so to form a spindle. So the protein in bacteria does a different thing. It usually forms a ring, at the division site and then gets squeezed in. But it wasn't until we could have the ability to image them 3D using crystallography, X-ray crystallography, that a guy in Cambridge, MRC in Cambridge, or LMB it is in Cambridge, actually showed that the structure is remarkably similar. Even though at the DNA sequence level, at that point, no one really could identify that they were similar. But then, of course, with more sequencing, computing power with sequencing, then enabled heaps of different algorithms to show that actually there are some similarities, right? But the homology sequence is not very high. But if you look at the 3D sequence, it's remarkable. Yeah. And their behavior is quite similar. So that was an amazing story to see over, 25 years or so. Yeah. Yeah.
So we can cope. So Liz,
yes.
tell me, what are you working on now?
So it's lovely to be able to now work with the younger academics, particularly research academics. I spent all my life, pretty much all my career, doing research. And yes, that's my main interest. I'm a very curious person, but we're also people. And so, in science, as with anything, there's different personalities, and I've experienced lots of different things. Colleagues defending their stories. I worked at Harvard, very hierarchical. There's huge egos there, as there are in many places in the world. And as a woman, I think also very different, because I would go to conferences and there would be me and a couple of women, but not many. And certainly at UTS, when I first came, there was one other female professor, one other. out of about, I don't know, at least 50 staff, 50 academics, probably more. So not a lot of role models. And I spent quite a bit of my career thinking why, that I was very different from everyone else, as you hear people say, right? And yes, we all have egos, but I was really, my key thing for me was to do the best science I could do. And what that means is that you need to sometimes realize how you are as a person and be willing to be open to possibilities of how to interpret something. We all have a bias, but so to me, that was really important. And then came the leadership positions that I had. And, in some universities, there was very ad hoc leadership where certain things happened for certain people and didn't happen for others. And, there was a travel fund apparently in one department that I never knew about, that various people were using. So it was just, I didn't feel part of the team sometimes. So that there were all these cultural things. And then as part of my learning about me and about how to be in the world, as you do when you get older, particularly, is I did a bit of mindfulness and self-awareness and starting to understand myself and my reactions to things, and therefore could understand a little bit more about other people's behavior. Not that it's bad, just different. And how to really, as a leader, put myself into someone else's shoes when I walk into their office, whether it be a boss or a student, whether they be happy or not, or whatever, right? So there's that thing about how you are as a person and just to be aware. So when that kicked in, my leadership skills were much better. And I worked with a strategic person who helped me with that, was define how does Liz Harry lead? And that's really what you need to do, is key into what you think is good leading. And of course, what you think is the right thing to do. So keying into core values. So what I do with outside opinion is in my job is I am able to, even with grant reviews and fellowship reviews, really get at somebody, really create a rapport with someone, and then see how we can excite the reader. And we do of the grant, and so they get a good assessment, because you need to assess it. Most of these people that I am working with, they're very clever and they're good scientists and good researchers. However, they haven't necessarily put themselves into the picture in a way that says, I love what I'm doing and I'm good at doing it, and let's tell the world that. Yeah, so that's a big thing for it. I also love the part of just thinking critically about science. If they're going to do this, do they need controls? Is this the best question to ask, and why? Why are you doing this? What's it going to bring? And don't stop at perhaps it'll do this. What else could it bring? What else? What else? What else? All the time opening up. And this is really helpful for people, particularly when you really don't know them. So you're just like a mentor coming in. So that's just wonderful. And I love changing. I love expression. So whether it's written or verbal, I love expressing words in a way that sit well and excite people. Yeah, it's wonderful to find those words. So when we have a Zoom after a grant review or a fellowship review, it's fabulous to talk to people and to just see their faces and the love of science that they have. They are extremely curious. They want to discover things. They're under a lot of pressure to get money, to get papers out. And we just make sure it's a bit of fun and excitement about what their passion is, yeah? Because if they don't have the passion, it's too hard. It's too hard. So the same with the workshops. What did I experience when I was younger? Are they experienced what? So what are the difficulties? They won't always be the same, but I can tell you most of them are. And a lot of the younger people are told what is needed of them. And they feel, particularly in this environment with universities not having a great budget, that they're told they need certain things. You need to get a fellowship, you need to do this. But they're doing it because they're told they need to. And so one of the things that's really important is to say, well, why are you doing this? And often it's because my supervisor said so. But then the real reason, then is, do you want to? Do you want a fellowship, really? Most of them do, but then they work out why. Where will it bring them? What is their goal? What's their aspiration? Yeah. So it's about inspiring them to aspire to something that they think is really terrific. Yeah. And I think that's most important. The research, of course, it's important to talk about what's possible for doing things in research, technologies, those things. And of course, I encourage them to talk to whoever it is that they think is the best person in the field. But they really need to get their mojo happening before anything. So for me, interestingly, that's probably comes before the science for me, or before the research and the critical thinking. It's, where are these people? How do we G them up? And the workshops are an absolute delight. And I see some of those people a year later. They talk. I give them. They workshop stuff. It's really a workshop. It's not a series of seminars, and I can give them advice and I can tell them what my experience is a little bit. But it's then they talk amongst themselves. And at UTS, they put many disciplines together. It's a pan-discipline, pan-UTS workshop. So that is fantastic. So someone from business can talk to someone from science. Someone from transdisciplinary can talk to someone from health. It's fantastic because they come up with ideas and then you can hear them going, oh, well, I was thinking of doing this, but then I could match this with this, couldn't I? And the other person is just, so they feed off each other. And each of the faculties give, they have different cultures, some of them the same issues, some of them different. But in the end, they share those issues. Yeah. They share the problems. They share solutions. What do I do if this happens? Oh, just do that, So it's this camaraderie that we're really bringing with UTS, quite a small university. So we bring it across the university. So, I applaud universities that have these sorts of programs. It's fantastic.
So it's sort of, you're talking about community and mentorship and across disciplinary communication.
Yeah, yeah. The last project I started before I left full-time academic work was actually a full, a true transdisciplinary project. And people throw the words multi and transdisciplinary around a lot, but really, biochemistry and molecular biology are really the same discipline, if you like, but slightly different. But really, mixing disciplines together is great. From a research perspective, it's extremely powerful. If you want to, identify a new medical device, of course, if you don't have the consumer involved, then, you could end up not selling a thing. You have the doctor involved, you have community people, just to say, if you had diabetes, would you use this? And you use the prototypes with them, so you work with them. And then you might work with a social person or a psychologist who will think, well, how do people think about this? Even before you start a project, actually, you do all of this, right? And then what about an education person? What about a business person or a lawyer? Would there be any legal issues with this? And design, right? Of course, design. So, and you always find, if you find the right people who are open, you'll always find that there's a space and there's a place at the table for all those people. And while it's very challenging, it actually makes it a great project because you've opened it up. In fact, we changed what we wanted to achieve many times, and it was very challenging because I went in thinking I want to achieve this. And then when everybody said, well, actually, that's not such a problem, this is a bigger problem in this broad context. And I thought, oh, okay, it was really difficult to do that. But if you really want to find a solution for something, or do some basic science that might have some application, it's really good to talk to people first because they'll have ideas about an application or ideas about how you might go about it, even from a basic perspective. Yeah. And then when you talk about the practicalities of doing research, how to manage, how to lead, how to work within the university system, it's a big public service institution. It's not easy. And you've got, there's lots of bureaucracy. There has to be to some extent. There's lots of forms, digital interfaces, platforms. They're always changing. And as a researcher at university, you are doing more and more of that type of work. You're spending a lot of time with biosafety, with ethics, with everything for good reason. But the world is turning a lot into making people the liability of stuff has become really important. People have to
Mm.
be hugely careful of that. So there's a lot more to do. So everyone complains about all the other stuff they have to do and not the research. So part of working across disciplines is to keep the motivation up and to help people solve. And every university has this, but when you get people together, they go, "Oh yeah, that's a workaround." So sometimes you have to just do it differently. Um, and yeah, there are times when you have to break rules that are not a big risk, but you just have to go around things sometimes because otherwise you go mad. Yeah. So that's what's great about getting people together.
Yes, and it sounds like it'd
Yeah.
be one of the advantages of, I mean, having a university at all where you've got all these different disciplines. So you've got the people together in their common disciplines, but then you've got all the separate ones, not
Yes.
physically very far away, and that you
Yes.
can easily get together and talk about all your different challenges.
Yes, I think like many people in the working world, you go down a rabbit hole and you stay there, particularly with research because it is very specific. That's why many academics find it hard to talk broadly to the lay person because they're like this all the time, right? Um, so yes, it does bring people out to communicate and it brings them out of their space. So you're often in the lab or mainly at the computer once you finish your PhD. You tend to do all the managing things and the writing of grants and whatever. So once they're out there in this workshop, um, yeah, they're fully present for about 2.5, 3 hours, and that's a good time these days. So you've got to make sure with a workshop when you prepare, because preparation's almost everything, that you are going to have, and while you're doing it, it's very fluid. A workshop, a good workshop is looking, seeing how people are, seeing that everyone's involved and thinking and giving their 2 cents worth, or $50 worth, are very worthy, um, and crunching the brain, really, really working to be open to what's possible. Yeah, so that's what happens, and then they go back to their space. So it just gives them a space to be out, and I keep encouraging them to do that. So we actually develop a research plan. So it starts with core values. What's important to you? That's a really interesting one. What does a mentor mean? What does a mentee mean? What do they do? Because they have a mentor through this process of workshops. And then we end up with a research plan and they give a talk about that. So um, part of that research plan, we encourage them to have a coffee with so and so every Thursday morning or once a month, or just to talk about what you're up to, someone who actually inspires you. It could be your mentor, but it could be someone from business to design or health um, to law, whatever. Just someone that you can connect with. Yeah.
It sounds like it's something that everybody should be doing.
Yeah. Yeah, I used to do it a bit too because I would get out of science and often, funnily enough, and I think this is probably the case for many
Mm.
work areas, is once I went into another faculty or I would often go to the UTS social events or, whatever. We'd have little events where we'd have drinks and celebrate or whatever, just to get out and meet people. Even if you don't talk research, you don't even have to talk shop. You can just say, hi, yeah, love working with UTS or love doing what I'm doing because, and people would be far more positive if you did talk about your work. There would be a little bit more sort of interest, if you like,
Yes.
and positivity around it. But yeah, and yeah, it sounds like my workmates were lovely. There's nothing wrong with them. It's just that when you talk to someone else about your work, it's the same in talking to someone on the street and you say, well, I'm a researcher and I studied honey for a while and its
Mm.
antibacterial effects and people are just so interested in that.
Yeah.
As soon as eyes glaze, you must stop. But, or even before that. But yes, keep your answers short is my advice, which is not what I'm doing now. But it's really important to get out. Yep. And get out to the public. I mean, I would say that public speaking for me still is more satisfying than it is talking at a conference because at a conference, there's still very much competition. Everybody's watching everybody else. Um, and it's just a different culture. It's changing, but and it can be really collegial as well, but it feels sometimes quite threatening.
Mm,
It has in the past. And so when you just get out and talk to people, they're just curious.
mm.
And yeah, it's really nice.
Oh, that's really good. Because
Yeah.
I don't know, there don't seem to be as many public talks since COVID as there used to be. University ones that you could go along to and hear a lecture from, or a talk, published a public talk from a scientist. So it's,
Yeah. Yeah, I think that's a shame. I was just reading in the paper, I think it was the Sydney Morning Herald last week, because International Women's Day was on Saturday. There were three women who talked about radio broadcasting in a different way. And it was so refreshing. I'm going to listen to some of them are just doing podcasts because they don't have enough money to go live 20, all the time. So, but they're great. And there are different ages. One of them's 26, one of them's in their 40s. And they're talking about things that are relevant to not just women, but yeah, sometimes women. And there'll be other people like them that are just concentrating on partly what they find is missing. And I think that's great. I was actually really wanted to get in touch with them because I think about science communication quite a bit. And if I wasn't a researcher, I probably would have done communication, science communication, because it's just so important to get those stories out there. And there's nothing like when I listen to a great podcast or a great story, it just lifts me so much, because it's not just about what that person's talking about, it's how they are. Um, so I listen to a lot of First Nations podcasts, and they're just, I can never, I'm astounded by their kindness and generosity and open-heartedness. So that brings me great joy, particularly currently. Yeah, with all the other things we're hearing about in the news. Yeah,
indeed, it is
yeah,
good to find kindness and generosity and
yeah,
curiosity in the world.
yeah.
Most people have those things, and it's good to
They
be
do.
reminded.
They do, and I think researchers have to be, I do think researchers do need to be reminded that they are many academics are introverts, that is well known. It's not a problem with that. But having a lot of introverts together or a lot of extroverts together, they tend to be similar to each other in those ways. And that can sometimes, that's another reason why people need to get out and about a bit, just to mix around a bit,
Yes.
um, because it can become a bit locked in certain ways. That's all. It's not bad. It's just,
Mm.
it's not always the best culture is a diverse one, of course.
Seems to me that all the things you're talking about here, uh, it's a part of the scientific career that isn't done in classes in your degrees, and it's almost like an apprenticeship, a mentorship that people learn this side of the career. Is that right?
Yeah. Yeah, so I just think more and more that the bottom line in a successful life is about just who you are. And, this may sound cliche, but just loving who you are. I think that's where a lot of our uncomfortability comes from. And because of that, there's the behavior that comes from that that's just less productive and less enjoyable. Yeah. So
Hmm.
often people hold on to certain things because they don't want to let it go. Um, but yeah, I think I'm just thinking about what you would introduce to students. And I think, in schools, for example, and I don't do a lot of work with schools, it's something I would like. They're often doing things like ethics in schools and, thinking about different problems and getting to know each other in that space is really important. So, so some sort of, I don't know what the social science of science, how do we work together? There's probably work in that space. Yeah, I'm interested in that, your question. I'm not quite hitting it, I don't think, but
That's all right. It's something to think about,
Yeah,
maybe.
yeah, yeah.
I know there's, I haven't read it amongst my books that I carefully packed when I moved here. There was a book on the anthropology of science called Down Among the Scientists.
Ah,
I haven't had a chance to read it, so I can't tell you much more than that at the moment.
right.
But I know somebody
Anthropology.
looked at that.
Yeah.
But of course, that's not quite the same as being able to do it.
No, but actually, a dear friend of mine, actually, when she was much younger, was working with an anthropologist. She was a chemical engineer. She was working with an anthropologist in terms of engineering education. So
Mm,
it's a little bit related to that. Um, yeah. I think the history of science is very interesting in terms of how people behaved.
yes.
And it seems bizarre, but actually, I've seen some of that behavior now
Mm,
where people are protective of things and whatever. Um, I guess what I feel strongly about is that we need to think about what changes are worth doing. And I mean doing, not just thinking about. We need to think about how we can act better to enable the younger scientists to actually lead us um, in their thinking, in what they see in the future, what concerns them. We don't really. Sometimes we have them sitting on committees where they do talk, but we don't really get at the detail. I met with the PhD students. Well, I did many times as a director of an institute, a research institute. And what I asked them basically is, when you finish your PhD, what is it that you want to have achieved? Yeah. And there were six things that I remember. There were six or seven things. Um, and I don't know the answer to that question unless I ask them. It sounds a simple statement, but actually we so assume so much. Yeah, we assume so much. So one of them was, I want the world or the people, I want society to know what I've done and why it's important. Even if it's just the basic of scientist, it doesn't matter, because if you don't have basic science, you never have application, you never have pharmaceuticals, you never have medical devices, you never have understanding how we eat good food or we have microbes in our gut, right? We never understand disease. So it's so important. We seem to forget that. So basic research is relied upon in society for so many things, good and bad, sometimes. Um, so losing my trail of thought now.
you're talking about what people wanted to get out of their PhDs,
Oh, right.
importance
Yes.
of knowing what you want and who you are.
Right. So the other thing, apart from communication to it, was to um, to have known that they've done the best science they could.
Mm,
Um, what were the others? Um, to be acknowledged for what they do. And
mm,
there are a few others I can't remember, but they weren't things I could have guessed. And most of the academics that I know, particularly in science, I know science very well. They don't love talking to the media. They don't necessarily know how to talk to the industry. Again, it's not a judgment. But if they have students and postdocs in their group, how on earth are those people going to do that or be encouraged to do it or have confidence to do it, right?
mm,
So we need to be open with our lab groups that we tend to say, this is my group. I've got 15 or 25 or people. Yes. And they're doing great science. But do we really know whether they're totally doing their best and can be at their best? Are we allowing them to be at their best? And I think in any work area, that's what we need to ask them, or in whether that's the direct question or not. But they need to feel they can do their best in this environment. This means they need to be trying to be as healthy as possible, as happy as possible. And what does that require? Yeah.
mm,
Are they doing the best science? Are they happy with what they're doing? Are they happy with their supervisor and the responses they're getting? Are they happy with the culture of the group? Yeah. And this is really important. And clearly not everybody is in.
Mm.
So this is the key in finding out what do PhD students need, what do postdocs need. We also have a postdoc society. So our institute started this and then it took over in the faculty, and they still have these groups. I think there's also a women in science group in the faculty. There's a ECR, early career research, which is 5 years, within 5 years of PhD. They are these groups and they're becoming strong and they're becoming a confident group of people. And we've also employed, over the time I was there, we employed some new women to come in who were, rocket scientists, and lovely and supportive. So yeah, I think we need to be open to say during this process we need to be open to what we might be doing wrong ourselves. So the best and hardest thing in leading is to be able to get that feedback yourself if it's negative, right? And I did in that case when I was encouraging labs in their groups. I was encouraging the younger people to talk to their supervisors, and a couple of my postdocs came to me and said, a lot of stuff's working, Liz, but there is one thing in this lab that's not working. So they shared it with me.
Mm.
And I wasn't keeping an eye on a couple of things, right? And I felt really, it was so hard to take. But I asked for it and I got it. And so I understood what it felt like. And that is really a gift because I understood how hard it is, right? But unless you can do it, you're asking people to do stuff that you can't do, you know?
Mm.
Yeah. So that was a huge learning process for me because I then changed it and we got it better. And they found it so hard to tell me. So, and I'm pretty approachable, which they all tell me. So it's, those sorts of things need to be out there and they need to be voiced in various ways. Yep. And they need to change. Yep. One example, when we had, I was chair of the honours committee years ago at UTS when I first arrived, and we have various written assignments or written feedback for their essays and various things. One person just wrote a whole lot of things, just a whole lot of things that was wrong with it. It was like 3 pages of stuff. Another person wrote no, yes, no for feedback. So
Yeah.
I talked to them both separately and said, this is very hard to get this from a student. Students often would come to me and say, this is not helpful.
Mm.
So one of them said, well, this is the sort of feedback they're going to get in the real world.
Mm.
And I thought, oh, what a shame that they're accepting that the real world is going to keep doing this and that it's okay.
Yes.
This to me is just, oh my God, it was an awful feeling. I thought, oh no, this is where we're at, So this is what I'm talking about with don't accept that this is okay. And it's not easy to buck the system, but if we can get the younger ones, it might take a while, but once they get to that stage, at least they'll be a bit more bolshy and a bit more standing up for themselves and
Mm.
standing up for the culture and saying, hey, this is not okay. We shouldn't be doing this. We should be doing it this way. What does everyone think?
Mm.
Learning how to communicate that properly. Yeah.
Now,
Yeah.
I have to warn you, we're down to the last 5 minutes of this
Yes.
session.
Yes.
So if you'd like to make a concluding statement, we can do that now. Or what would you like?
Yeah, we can do that now.
All
Yeah.
right. So do you have a concluding statement, Liz?
I guess I think the important thing about science is that you just do the best science you can, do it truthfully, so that at the end of the day, you know that you've done your very best to find out how the world works. If you're curious, which you will be when you do science, you will want to know the truth and do whatever you need to do to find the best people to work with, the best supervisor, not just a good scientist, but a person that you feel comfortable with, that you feel you can be open with, that you can ask any question that is not stupid. No question is stupid. And just don't put a fence around yourself. Anyone can be good at what they do. It's just a matter of letting go I was inhibited earlier in my life thinking I couldn't do it. And that just made me less successful. But then I became successful anyway by just letting go. So failing's important, most important, and you will fail if you're going to be one of the most successful. Find people that inspire you. Find places that you're happy to work in. If you are not, do not waste your time. There's always a place for people who want to be their best. So thank you.
Thank you, Liz. That was amazing.
Thanks, Ian.
I really appreciate it.
Study science, because you will find in the study of science a richer, more rewarding life.